Hi everyone, thank you very much for joining us today. So the Chappel Memorial Lecture Series was established in 1987 through an amendment from the Ontario Veterinary College graduate Dr. Clifford Chappel and his wife in memory of his father Herbert James Chappel. And I want to precise that we have the honor of having Dr. Clifford Chappel online. So thanks a lot, Dr. Chappel, for joining us. So Dr. Clifford Chappel had an exemplary career in medical research. His most remarkable achievements, among a lot, have been in identifying and in some cases stopping the harm to public health caused by dangerous pharmaceutical, environmental, and biochemical agents. Held annually since 1988, the Chappel Memorial Lecture Series has hosted numerous internationally known scientists who have inspired University of Guelph graduate students, faculty, and staff to achieve the highest possible goals in basic or applied research and to motivate undergraduate students to consider alternative career paths and become leaders in their profession. So now I would like to introduce Peter Hansen. He's a distinguished professor and Ellie Red Larson Professor in the Department of Animal Sciences at the University of Florida, where he has worked since 1986. Working primarily with the cow, his research focuses on the physiology and genetics of early pregnancy and terminal biology and development of methods to improve fertility, enhance assisted reproductive technologies, and develop genetics and physiological strategy to reduce the impact of heat stress. Key advances from his work include the use of embryo transfer to alleviate infertility caused by heat stress, development of dairy cattle that are genetically resistant to heat stress, and manipulation of preimplantation development programming to modify postnatal phenotype. Dr. Hansen received a BS in Agricultural Sciences from the University of Illinois in 1978 and the master's degree and Ph.D. degrees from the University of Wisconsin in 1980 and 1983. He did a postdoc fellowship at the University of Florida from 1983 to 1984 before joining the faculty of Florida as an assistant professor in 1984. We actually had the pleasure to host Dr. Peter Hansen for a sabbatical leave in 1996 or
93.
93. So Peter shared with us that he had fond memories of his time at OVC. He's a fellow of the American Association for the Advancement of Science, American Dairy Science Association, and American Society of Animal Science, and distinguished fellow of the Society for the Study of Reproduction. Among his leadership posts, he has been president of the American Society for Reproductive Immunology, International Embryo Technology Society, and the Society for the Study of Reproduction. So with my French accent, I'm sorry, I hope that you understood everything. I'm very honored to introduce you to Peter Hansen.
Yeah, no, thank you. It's a great honor. I was very honored when I was sent the email asking me to give this seminar. I thank Stephen LeBlanc for help getting me here. I thank Dr. Chappel for his generosity, really, in continuing this tradition of education that OVC is part of. It's very humbling for me to get to participate in that education through this lecture. And the title of my presentation was really based on the idea, rather than just focusing narrowly on some of the research objectives I'm following, was to look broadly at where I think the topic of assisted reproduction in cattle is going. And I thought it was particularly appropriate to do that here at Ontario Veterinary College, because at least when looking at assisted reproduction in cattle, the University of Guelph has played an essential role in the development of technologies for in vitro production of embryos. So I want to just share with you some of my ideas about where we've been when it comes to assisted reproduction and where I think we're going. So I'm going to really talk about 5 revolutions in assisted reproduction in cattle. That image there comes from an online magazine, The Bovine, which is, you read the bovine? Yes, it's for dairy breeders. They have a lot of colorful cartoons. And I'm going to talk about 5 revolutions that have really changed the way cattle reproduce, and as a result has advanced our ability to select cattle genetically for superior production traits and health traits. So one of the revolutions is a pretty old revolution. Artificial insemination was developed probably as early as 1902 in North America. Sex semen is really a spin-off of artificial insemination where it became possible to select X or Y sperm so as to control the sex of the embryo. That's a revolution that began in the 1980s, but really didn't start to affect how cattle were bred and selected genetically until the advent of genomics, which I'll talk about in a minute. Similarly, embryo transfers, kind of an old revolution. 1950 was the first embryo transfer in cattle. But again, beginning after about 2009, the realization of the promise of embryo transfer began to be fulfilled after genomics. And then I'm going to talk about 2 other technologies. Cloning, it's a very controversial technique in animals, developed in the 90s. And then stem cell technology, which I think I'm really going to take you to the future, I hope. I think this represents the next advance in artificial assisted reproduction in cattle. So this is, believe it or not, a very famous photograph, probably none of you have ever seen. At the time this photograph was made, this was supposedly the most photographed bovine in the world. The first calf born from artificial insemination in North America, supposedly. I think, in fact, cattle have been born by AI since about 1902 in the US. But this was the first calf born with kind of the modern techniques of artificial insemination that are used today. Transvaginal, transcervical deposition of semen into the cow. And this was a very controversial phenomenon. A lot of people thought this was fooling around with God's plan for reproduction, and human beings should not be getting involved in the process. A lot of people thought that calves born from artificial insemination would be deformed, would be monsters, because this is not how God planned it. In fact, this first artificial insemination project in New Jersey, the first 10 calves were all bull calves. People said, see, something's wrong. And even 10 years later, people were still concerned that calves born by artificial insemination were abnormal. So this is a poem from Hoard's Dairymen in 1949. I'm a little heifer, I'm an artificial calf. Some people think it's funny, but go ahead and laugh. When I grow up to be a cow and join the milking herd, I bet I'll be a big success, perhaps the latest word. So artificial insemination really transformed the dairy industry in particular, the livestock industry more generally. For one thing, it was no longer necessary to keep bulls on the farm. The dairy bull was the leading cause of farmer fatalities in the first part of the 20th century. Bulls are very dangerous, as this ad in Hoard's Dairyman points out. Is the dairy bull safe? Look in the rural graveyard for the answer. And I found these pictures because I'm kind of interested in history. Back in the Depression, the United States government paid artists and photographers to go out and record life in the United States, kind of as a way of increasing employment. So this guy in Idaho took these pictures of a bull stud in Idaho in 1940. So this is how they used to bring in advanced genetics onto dairy farms before artificial insemination. You can see the herdsman with this big long pole trying to grab the ring on the bull. And they load the bull up into a trailer, take him to where the cow is, breed the cow, and then wash the bull's sheath to avoid venereal disease. It's a big process. So artificial insemination eliminated all the danger of working with bulls, all the difficulty in achieving planned matings of cows with bulls, but most importantly, greatly increased the genetic progress that was possible from planned matings. So with artificial insemination, it was possible to produce multiple offspring from individual bulls, measure the performance of those heifer offspring, and use that information to identify which bull was genetically superior. So typically in an artificial insemination stud, they would buy bulls, produce about 50 daughters from each bull, measure milk yield on the daughters, and identify the best bulls. And then because artificial insemination allows the production of thousands of units of semen in a year, only the very best bulls could be used in a genetic selection program. So, I mean, we had bulls like Elevation, probably the founder bull of the modern Holstein breed in North America. In his lifetime, over 500,000 straws of semen of Elevation were sold. Just about all the bulls in North America descend from Elevation. But more importantly, I mean, the genetic progress associated with genetic selection was greatly increased by artificial insemination. So just look at these data here. This is milk yield per lactation in pounds or in kilograms. And you can see in 1957, the average cow in the United States of America produced about 13,000 pounds of milk per lactation. In 2022, the average cow produces more than twice as much milk, 28,988 pounds. Probably half of that improvement in the amount of milk produced per cow per year is because of artificial insemination, because of the increased genetic selection possible with that technique. And I actually put this slide in for Christine Bays, who's very interested in greenhouse gas emissions. Probably the best way to reduce the carbon footprint of an animal used for food production is to increase its productivity. So if you look at kilograms of CO2 equivalent produced per kilogram of fat protein corrected milk, as cows produce more milk because of genetic selection or whatever reason, the carbon footprint goes way down. So, you know, as a result, if you look at greenhouse gas emissions per kilogram of food product, milk is pretty low. Actually, less CO2 produced than produced for rice or almonds. So this genetic selection to produce a cow that can produce more milk is important not only to reduce the cost of milk and increase its availability to consumers, but also to reduce the carbon footprint of milk production. The next big revolution in assisted reproduction in cattle was sex semen, which was originally developed by Dr. Johnson at the USDA in the 1980s. I'll talk more about this in a little bit, but sex semen made it possible to select the sex of the offspring. A Y-bearing sperm cell has about 3% less DNA in it than an X-bearing sperm cell. And if you label the DNA in sperm with a fluorescent dye, you can develop flow cytometers that will allow you to distinguish between the X-sorting sperm and the Y sorting sperm. And with the kind of flow cytometers that are available now, X sorted sperm, which produce females, can be separated from Y sorted sperm, which produce the males, with about 90% accuracy, so that you can control the sex of the offspring as a result. So the next revolution I want to discuss is embryo transfer. Why can't we do on the female side what we do on the male side? So artificial insemination made it possible to produce literally hundreds of thousands of offspring from an individual male. Embryo transfer is an attempt to do a similar kind of thing at a lesser scale on the female side. The first person to successfully achieve embryo transfer in cattle was Elwyn Willett. I got this slide from one of Dr. Bettridge's papers. He later died of a brain tumor when he was still in his 30s. But he was the first person to successfully transfer an embryo from one female to another female. And this technology was improved through multiple rounds of technological advances until beginning in the 1980s, it became possible to produce embryos in the laboratory by so-called in vitro fertilization or in vitro production of embryos, where oocytes or eggs are collected most commonly from living cows using an ultrasound-guided needle. Those oocytes are matured, allowed to complete meiosis 1 in culture. They're then mixed with sperm. The sperm have been capacitated, modified biochemically so they're capable of fertilizing the egg. And then fertilization occurs in vitro. The embryo is cultured for 7 days and then transferred into a recipient female. With this kind of technology, oocyte harvesting can be performed as often as twice a week, but probably more commonly every 2 weeks. So 26 times a year, with the average of about 5 embryos produced per procedure. So theoretically, you can produce 100 embryos per cow, which is a lot less than for the bull, but still a lot more than with natural reproduction. This procedure is routinely practiced on some artificial insemination companies on prepubertal animals and on pregnant animals till about 4 months of pregnancy. So as compared to the original superovulation procedure of Willett, in vitro production of embryos magnifies the number of females or number of embryos that can be produced from a genetically elite female. And as I mentioned, a lot of the work that was done to optimize procedures for in vitro production of embryos was conducted right here at the Ontario Veterinary College. So I just have pictures here. Alan King is in the audience. Keith Bettridge is in the audience. Also Stan Lebo, who did a lot of important work on cryopreservation of embryos, was a faculty member here at OVC. And Don Rieger was a faculty member here. Here's a little paper I'd encourage everybody to read that Keith wrote, Thereo-Ontology, a personal view of 40 years of farm animal embryo form and function. So when I, as an associate professor, was trying to learn how to do in vitro fertilization so that I could study the effects of heat stress on the preimplantation embryo, like Isabel said, I came to Guelph. Here I am at the University of Guelph, working in Alan's lab. Here I am experiencing Canadian culture at its finest at the Fergus Highland Games. So, I mean, that was a personally very rewarding experience for me and really changed my life as a scientist. So what changed the life of assisted reproduction was genomics. So in 2008, the first bovine genetic chip was developed, the Illumina SNP50 chip. So this allowed, for the first time, animal breeders or geneticists to genotype animals, to understand the genetic merit of individual animals without the need of measuring their actual performance or the performance of their daughters. So like I mentioned before, in the old days of artificial insemination, bull studs would produce about 50 daughters from each bull, measure the milk yield of the daughters, find out who was the best bull. So that took about 5 years. With genomics, it's possible to identify genetic merit for the trait of interest as soon as you can get DNA from the animal. So when it's born, when it's a fetus through amniocentesis, or even when it's an embryo. So it completely changed the use of technologies like sex semen and embryo transfer. Before the SNP50 chip, those techniques were not really well utilized because of the inaccuracy identifying genetically superior females. But after the chip, it became possible to find the best females with almost the same accuracy as you could find the best bulls. And so now, for the first time, it made sense to do things like in vitro fertilization, to produce offspring from the best females. And all of a sudden, sex semen became an important tool. Because if you can identify the best 100, let's say you can identify the best 50% of the heifers on your farm, you can separate out which cattle will produce the replacement females from which cattle won't. You use sex semen to produce heifers from the best top of the herd, and then use a different type of semen. These days we use a lot of beef semen to produce offspring from the lower part of the herd that will not produce the replacements for the next generation. So since genotyping, sex semen has become a very widely used technology. So this is just kind of a corny slide here. But before genomics, we found out who the best bull was by producing lots of females from each bull. And we never really knew who the best female was because we only had a record from her, maybe one or two of her daughters. But after genomics, we could use DNA technology to find out which was the best bull and use him for artificial insemination. And we could use genomics to find out which was the best female and produce multiple offspring from that female using embryo transfer. So every year, the International Embryo Transfer Society tries to estimate how many embryos are transferred in the world. And I'm sure this number is a great underestimate because a lot of people who do embryo transfer don't report their results to the International Embryo Transfer Society. But take a look. 2008, the SNP50 chip was introduced to the world. The number of in vitro produced embryos transferred in that year was about 450,000. But after the introduction of the SNP50 chip, the utilization of in vitro production of embryos greatly increased, so that by 2019, a million embryos were transferred per year. So the combination of genomics and assisted reproduction has allowed for increased rates of genetic selection. I'm always looking at these estimates of which are the best bulls for my own personal reasons, because I'm trying to mate cows to bulls. But if you look here, this is just a summary of the top bulls in the Holstein breed in April 2023, the genomic young bulls. So these are bulls that are identified as the best just based on their genomic information, not on any production data. And then all these bulls have names: Ladies, Manor, Smoke, Owen, ET, so on and so forth. Every bull that has ET ending in his name is a bull that was produced by embryo transfer, probably using an in vitro produced embryo. So there's 42 bulls on this list. 37 of those bulls were produced by embryo transfer. So now at the elite levels of the Holstein breed and other dairy breeds, almost all of those animals are produced using embryo transfer. All right, now I'm going to kind of talk a little bit about where I think these technologies are going. Just going to say a little bit about cloning and also something about stem cell technology. I think these are I say cloning's a revolution. It was a revolution back in the 90s. I think you're going to see increased use of cloning in the future, and I'll try to explain why. And then stem cell technology. So these first three technologies I talked about are all pretty much achieving what occurs in vivo artificially, in vitro. But these next two technologies are really like paradigm shifting technologies because they're technologies that fly in the face of the germ cell theory. So back in the 1800s, before people knew what a gene really was, people were trying to understand the biological basis of inheritance. And August Weismann came up with the germ cell theory. So Weismann said, and he was right, only germ cells transmit genetic information to the next generation. So that was kind of controversial because at the time Lamarck said, any cells that experience some environmental changes carry that on to the next generation. But Weismann said no, that all the genetic information received by an individual organism comes from germ cells. So the genetic material in the germ cells contributes to the next generation. There's genetic material in other cells, the somatic cells, but that genetic material is never passed on to the next generation. So you can experience a mutation in the liver, and that might have profound effects on the function of the liver, but that mutation cannot be inherited by the offspring because all the genetic material comes from the germline. And there was a barrier, an insurmountable barrier between the soma and the germ cells, so that genetic material does not pass from somatic cells to the next generation. So that barrier is now being broken. And it was first broken by cloning, somatic cell nuclear cloning, where Ian Wilmot and Keith Campbell and others took a mammary cell nucleus, put it into an enucleated egg, and produced a new organism. Little Dolly the sheep, right? So what they did in that procedure, this is a mouse, but it's the same principle, was remove the genetic material, the germinal vesicle from the oocyte. Ordinarily, that pronucleus from the oocyte fuses from the pronucleus of the sperm, produces a new organism. They removed that germinal vesicle and placed, so now there's no nuclear material in the oocyte, and then they placed the nucleus of a mammary fibroblast. And that DNA then drove development of the new embryo. So now DNA from a somatic cell gave rise to a new organism. So this, for good reason, got a lot of attention. The problem is There's a lot of epigenetic reprogramming that occurs during natural fertilization and early embryonic development that allows or that removes all of the epigenetic marks from the oocyte and sperm and then puts new ones on, new appropriate ones that allow embryonic development to occur. So that doesn't exactly occur during cloning because you're taking a nucleus from, say, a mammary cell that's epigenetically a mammary cell and using that to support development. So the process of cloning is very inefficient. These are some data from the 1990s, but I don't think the results have really changed that much. So if you just look at skin, in this experiment, they did 1,103 little cloned embryos. They ended up with 4 calves. So it's a very inefficient process. And even if pregnancy is established, many of the placenta develop abnormally, can develop so abnormally they kill the cow. And then many of the fetuses are born abnormally. So I think the initial excitement over cloning kind of dissipated when people could not overcome this problem. That the efficiency of the procedure was too low to make it practical, and then the negative consequences of cloning were pretty dramatic. But I think cloning is making a resurgence. That our ability to understand how nuclear material is controlled epigenetically is greatly increased as compared to the early days of cloning. And I think you're going to see more research on how to epigenetically regulate the cloned embryo so that it has a higher pregnancy rate and less frequent incidence of abnormal fetuses than is currently the case. And probably the most aggressive company in the world with respect to assisted reproduction technologies is ST Genetics. Used to be called Sexing Technologies. They routinely use cloning in their bull production programs. So here is another ranking of the top genomic bulls. This is from 2021. And if you look at the top 3 bulls, one bull named Captain, another bull named Jack, another bull named John, they have the same genotype because Jack and John are clones of Captain. And in fact, ST Genetics performs amniocentesis on selected pregnancies and uses the cells to clone the pregnancy. So even before the bull is born, his clone is already being produced. So instead of having one Captain whose semen sells for maybe $100 a straw versus $10 a straw for another bull, they have 3 Captains. And ST Genetics, if they have an embryo produced from a mating that they think is likely to produce a genetically elite animal, and the embryo doesn't look very good, they just take the cells from the embryo and use it for cloning. So I think you're going to see cloning, which I once thought didn't have much role in animal reproduction, is going to have a resurgence. So this is kind of where we're at now. You can use artificial insemination to identify the best bulls and distribute the genetics from those best bulls very widely. And then to a much lesser extent, we can increase the number of offspring from genetically elite females by collecting oocytes from the cows using transvaginal ultrasound-guided approaches, fertilize those oocytes in vitro, and produce embryos. So maybe we can produce 100, 150 embryos a year from genetically elite females inseminated with genetically elite bulls. And we can also take somatic cells, a fibroblast, other cell types, and perform cloning with low efficiency, but I think that efficiency is going to increase. So that's kind of the status of the art right now. So where are we going? So the next step in our advances in assisted reproduction is stem cell technology. So I keep screwing up my computer here. What are stem cells? I mean, the literature is full of discussion of stem cells right now. This is kind of my definition. The Play-Doh of cells. You can mold it into anything you want. They're undifferentiated cells that can give rise to one or more specialized cell types. And of course, their level of plasticity varies from cell type to cell type. So a one-cell embryo, a two-cell embryo, as far as the morula stage, 60 to 100 cells, each of those cells has the ability to differentiate into all the different tissues of the organism. Right? So those cells are really totipotent stem cells. But once the blastocyst forms, this is the first differentiation state of the embryo, the cells of the embryo are no longer totipotent. The outer cells of the embryo, the trophectoderm, are still stem cells, but they can only give rise to placental tissues. So they're pluripotent stem cells that are programmed to give rise to one or more of the cell types in the placenta. And the inner cell mass, which eventually becomes the fetus, is also no longer totipotent. It's pluripotent. It can give rise to all of the tissues of the fetus, but cannot give rise to the placenta. And then as embryonic development progresses, there are other populations of stem cells that exist in different tissues, right? Like in the bone marrow, there are stem cells for producing white blood cells. But they're much more restricted in their plasticity. They can only give rise to one or several different cell types. So today, people are using embryonic stem cells and adult stem cells to try to expand what we can do in terms of assisted reproduction. So let me just give you a few examples of that. So, what are we going to use if we want to make an embryonic stem cell? Well, probably the cells of the inner cell mass, because while not being totipotent, they are pluripotent and they can give rise to all the different organs in the animal. So people have been trying to generate embryonic stem cells that can then be used for several different purposes. So one purpose of the embryonic stem cell would be to use it for cloning. So you could theoretically screen 100 different embryos for their genetic merit and then produce embryonic stem cells from those embryos and use the stem cells from the very best embryos to do somatic cell cloning. There's a lot of interest in making artificial embryos. So you can take embryonic stem cells from the inner cell mass and regenerate a structure that looks like an inner cell mass. And you can take embryonic stem cells from the trophoblast and generate a structure that looks like the trophectoderm. And if you put those two cell types together, you can generate something that looks just like an embryo. It's got cells that function like the inner cell mass, cells that function like the trophectoderm. So can we use these artificial embryos to increase the number of offspring from genetically elite animals. Also, kind of what many, many people right now are trying to do is to get embryonic stem cells to differentiate into oocytes. Because if you can get an embryonic stem cell from a genetically elite embryo to differentiate into an oocyte, you can fertilize it with semen from a genetically elite male and increase the rate of genetic selection and increase the speed of genetic selection, because genetic selection is occurring in the lab, not after 9 months of gestation, et cetera, et cetera. And why limit ourselves to oocyte stem cells? Why not take stem cells from the embryo, turn them into oocyte stem cells, or turn them into sperm stem cells, and perform matings of oocytes and sperm derived from embryonic stem cells to increase the rate of genetic selection? So that sounds kind of crazy, right? But it's happened in the mouse, and people are trying to do it in the cow. This is a blastoid, an embryoid. It looks just like a blastocyst, but it's formed from trophoblast stem cells isolated from a cow embryo and embryonic stem cells isolated from a cow embryo. And these two separate cell types are cultured with different growth factors, aggregated together in a little V-shaped culture dish, and the cells spontaneously reaggregate to form something that looks very much like an embryo. So Carl Yang from University of Florida, this is one of his embryos, or embryoids. He's put these into cows, gotten the embryos to go at least to day 14 of pregnancy. So I'm not sure you can actually get live offspring from a structure like this, but maybe we can. I'm not going to go through this. This is a summary of progress in the mouse and in the human and the primate to develop oocyte stem cells or sperm stem cells from embryonic stem cells, or from induced pluripotent stem cells, which I'll talk about in a minute. So in the mouse, people have generated oocytes from embryonic stem cells, and people have generated sperm cells from embryonic stem cells. And they've identified primordial germ cell-like structures from embryonic stem cells in the human. So in 2018, Pablo Ross, who was at University of California, Davis, who's now at ST Genetics, I'm sure doing all of this stuff secretly, he was the first person to report a technique for making embryonic stem cells in the cow. Very easy to make embryonic stem cells in the mouse, pretty easy to make them in the human, but it was very difficult to make embryonic stem cells in the cow. But he identified a procedure based on inhibiting Wnt signaling that allows cells to be formed from the inner cell mass of the bovine embryo, and you can differentiate those cells into the three endoderm, ectoderm, mesoderm cell types. So for the first time, stem cells existed in the cow. And then he published this review article in Biology Reproduction where he speculated as to what we might do with these stem cells. So, and I just bet they're working on this. So according to Pablo, once we have embryonic stem cells that can turn into oocytes and sperm, we can develop an in vitro breeding system to greatly shorten the generation interval for genetic selection. So, Pablo said, you do in vitro fertilization with sperm from an elite bull and oocytes from an elite female. You produce a whole bunch of embryos. Then you produce embryonic stem cells from all those embryos. Test them genomically to see which ones are the best. Right? They all have the same sire, they all have the same dam, but because of genetic recombination, they'll all be genetically different from each other. So grow up the stem cells, culture the stem cells to see which one has the best genotypes, and then make oocyte stem cells from the embryo if it's a female, or sperm stem cells from the embryo stem cells if it's a male, and repeat the process. Take those new oocytes, those new sperm, produce more embryos, repeat the process. So something that takes probably 2 years right now, he says, you could do in 3 or 4 months. Depending how long it would take you to get an embryonic stem cell to become oocyte or a sperm cell. So we can go beyond that now. So what Pablo's talking about is taking cells from the embryo, turning them into embryo stem cell, and then either making artificial embryos or making oocytes and sperm cells. But now we can make pluripotent stem cells from probably just about any cell if we genetically modify the cells to overexpress transcription factors that are important for stemness. You know, the so-called induced pluripotent stem cell. Take 3 or 4 transcription factors, overexpress it in a fibroblast or another cell, and those cells start to act like stem cells. So if that's possible, you don't even need an embryo to start to make embryoids or oocytes and sperm cells. You can make an induced pluripotent stem cell, and then that cell can be differentiated into an oocyte or sperm cell, be used for somatic cell nuclear cloning, be used to make embryoids. And Vimal Selvaraj at Cornell University has succeeded in making cells that look very much like 8-cell embryo cells. So he took fibroblasts from cow, transfected those fibroblasts with these 4 genes that are commonly used to make cells think they're a stem cell. And then he also added a 5th factor, the simian virus large T antigen. Then he cultured the cells with different inhibitors of cell signaling. He added interleukin-6 or LIF to the culture medium. And he produced these cells that, in terms of their gene expression, looked like a 16-cell bovine embryo. And then when he placed those cells into this poor mouse, those cells differentiated into cells representing endoderm, ectoderm, and mesoderm. So he thinks he produced an induced pluripotent stem cell that could be used for some of these processes I've just talked about. So we've really come a long way from that 1939 bull calf born in New Jersey from artificial insemination. So I was a guest editor for a journal, Cabby Ag and Biosciences, which was a special issue on genomic editing. And the publishers of the journal, they produced this little figure, kind of this corny-looking cow, looking out into the horizon, right? The horizon of assisted reproduction and genomics. And you can see all these clouds, which are all question marks. So, I mean, there's a lot of question marks about all this technology. You know, where do we go from here? How do we use this technology? How will it change the nature of the cattle industry? But these same techniques can be used for any mammalian species you're interested in. How do we maintain genetic diversity? right? If one bull like Elevation produced a couple 100,000 offspring, the genetic diversity in the population declines dramatically. And just about all these techniques probably have as a byproduct reduction in genetic variance. So how do we maintain genetic diversity, if we need to, in populations of livestock? I didn't talk about it. I was gonna, but I didn't think you wanted a 2-hour Chappel lecture. There's a lot of epigenetic consequences of embryo transfer, of cloning, I'm sure of of stem cell technology. So how do we avoid those epigenetic consequences, those abnormal developmental phenomena that are associated with these techniques? You know, I was going to talk about gene editing, but also too long. All of these techniques involving the embryo and stem cells are very amenable to gene editing. I work with a company, Accelligen. They just produced a Holstein using embryonic stem cells and cloning that's got 4 gene edits in it. One for sleeping sickness, one for the SLIC gene, and 2 other disease resistance genes. So something that used to be really difficult, pretty easy now. So, you know, how will all these techniques affect our ability to edit genes? And then we talked about this at 2 o'clock. You know, how will the public accept these technologies? Like, I'm sure some of you right now are thinking, this all sounds pretty horrible. But, you know, it's going to happen. And so how will society view these technologies? I don't know, but I think I know. But I think they won't like them, but they'll have no choice. But I could be wrong about that. So with that, I'll be glad to take any questions. Thanks very much for your attention, and thanks for the invitation to be here.
I want to thank you, Peter, for coming.
Thank you. Thank you.
Thank you very much, Stephen, Carrie-Ann, and Katie, for organizing this Chappel lecture. Thank you, Dr. Chappel, for being with us today. And thank you, Kevin, and IT support for being here today. So thanks a lot, everyone. Thanks, Peter, again.
Thank you. Thank you.